Hello, everyone. Welcome to our Reclone community meeting, the third one of this year. And today, joining us, we have Ian. Uh, oh, sorry. You might hear my dogs, as always. They are always very excited about the meeting, so they're also saying hi. So we have our speaker today, Ian Kermadec. And Ian is a biochemist and a membrane protein specialist by training, having undertaken a PhD at Grenoble Alps studying the BMRA membrane protein. He subsequently worked as a research scientist and engineer at Counterculture Labs in Oakland, in the US, the Baltimore Underground Science Space, and the Learning Planet Institute in Paris, where he gained a lot of experience in using and escalating up production using Pichia pastori strain. He has since gone to co-found with his partners, Louise Lasselle, their own company, Enzymethic, which aims to develop enzymatic products that are license-free, sustainable, and accessible to everyone, everywhere. So a lot of outstanding work already. Thank you so much, Ian, for joining us, and
Thank you.
it's with you.
Okay, I will share my screen. Up, up. Sorry, my computer is not that fast. And poof, poof. Okay, so hi everybody. Thanks Yan Kai and Sibeli for the invitation. So today I will talk about Enzymethic. So this is the company that my wife and I found. So I will give a bit of background for why we created Enzymatic, what we try to do, and where we are basically. So for the where we come from, so I like Sibylle was saying, I did my PhD in Grenoble, and then we moved to Oakland, California, where my wife had a position in the Lawrence Berkeley National Labs. And me, I discovered during that time community labs. So I was a bit lost in what I wanted to do in the future, but I knew that I'd really love to do bench work. So then I found the place when I can do bench work and play around, and I was really happy about that. So I was introduced to citizen science when I discovered counterculture labs, and then I become more involved. I start having some board role, and I work in different projects, mainly real vegan cheese and open insulin. And then my wife got a position in Baltimore, and then we moved to Baltimore. So wanted to continue working on citizen lab. So I discovered there was a citizen lab called Bugs, and I kind of imported the Open Insulin project to Baltimore. So now Open Insulin have people working in Oakland and also in Baltimore. So I will talk a bit about Open Insulin because it's also really an inspiration to what we are doing now. So the goal of Open Insulin was to produce insulin in an open source manner because there is a crisis. I mean, it's still going where the price of insulin is really, really expensive in the US. And if you don't have insurance, then it could be a real burden and it can cause a lot of medical issue, obviously. So we still we want to produce insulin and find a way to distribute insulin in a different manner than what it's done right now. So the goal is to have decentralized cooperative who produce insulin. So I have a small visual to explain a bit more. I will not be too long on this, but basically the goal is to have a community lab developing the open source protocol and then having even insulin cooperative where in the board of the cooperative you have people with diabetes. So the goal is to be sure that the cooperative who produce insulin represent the interest of the patients. And alternatively, you can imagine having a hospital producing the insulin, or having some CMO producing the insulin as well. But the main goal is to have cooperative. So when we came back from the US, we wanted to continue working on the open insulin project. So there is not a lot of citizen lab, because the regulation is a bit different in France than in the US. So we needed to find a solution. So we still funded the open insulin the associations. We have Open Insulin France, but at the same time we needed a lab, and we were thinking, why not applying the philosophy of the open insulin project for the production of enzyme. So having open source production of enzyme and having cooperative who produce those enzymes. So we find a place in Nantes, where we are right now, and we now have a lab, an official lab where we can do GMO, which was kind of the difficult part in France. And we managed to find enough equipment now that we are functional. So finding equipment was also a bit of a hassle, because there is less free-flowing equipment in Europe than in the US. So we became some kind of auction experts, because it's a really great place to find equipment. So now we have a good station for protein purification with multiple ACTAs. We managed to find in an auction a nanodrop, a combo nanodrop spectrophotometer, so we can do most of the activity assay we want to do. We managed to get an up entrance that we plan to use for conditioning the product. And we also have a -80 for the storage of the strain. So we still need to find better equipment for the upstream process, because right now we have a really small shaking incubator, but for the downstream process, we are quite well equipped. So now I will talk a bit about what we mean by open source, because open source, I mean, it's like everybody knows, it comes from more the software world, and for biology, there is multiple aspects of it. And when people say open source in biology, that doesn't necessarily mean the same thing. So one of aspect that when people say open source, what they mean is open access. So there is no fee on scientific paper. Another aspect of open source is open information, that there is no patent or trade secret, and everything, all the processes are transparent and are free to use. And for biology, there is also the aspect of sharing materials, and this is the thing which often is overlooked, because it's not in the logic of patent. It's more MTA, and MTA have a different logic with it. So it's the agreement, transfer agreement. So I will focus a bit on the strain example, and to show what is our philosophy and what we are trying to achieve and trying to do. So for the strain, I will go back to when I was working with Open Insulin in Auckland, and we bought a GS115 strain on in vitro gen, and we worked with it. We were happy. We managed to have expression of casein protein, which would be useful for us for the processing of the pre-insulin. And 1 year after, we receive a mail, and in this mail, basically they say that when we sign, when we agree for in the purchase, was to use the PCR system for 1 year, and then if we wanted to continue to use it, then we had to negotiate with them to have some kind of license. So it was a bit frustrating, and to me, at least, kind of the eye-opening situation for me was that They continue saying that if we don't agree with an extension, then they can require that we destroy all the materials, and then we can basically lost a year of work on those with those trains. So then we start thinking that we have to, if we really want to have an open source process, then we need to secure the whole pipeline, and it also include the strains, because you can't work with some materials at one point having people saying, oh no, you can't use this anymore. And especially for the open insulin project, if we want to be able to share the strain, and if we want to be able to commercialize the insulin, then it could be a big issue if at one point somebody in the chain of production say, oh no, you can't do that because we don't want you to do it. So then we were looking at, okay, what other way we can find this Pisha pastori strain. So I start looking at the biological resource, but the issue with biological resource is most of them have MTA, which are not really friendly either for commercialization or for sharing the material as well. And so I found out that there is I think it's an association which is called the World Federation for Cultural Collection, and basically they have a website that I wanted to show here, but for some reason it was not working. So I just have the wiki page about it. But they basically it's a database of all the cultural collections. So there is around 600 and I mean there is on this registered by this association there is 676. So there is quite a lot, and you have a lot of different license. So it's really it's a big work to find out which biological resource have which kind of MTA, and maybe there is some there that have friendly MTA for sharing and for commercialized use. But so far with my research, the one I did on this, then I didn't really find any culture collection which have really good MTA for what we want to do with open insulin. The closest I found was some kind of some culture collection in India, where basically they say, oh, if you don't use it in the India territory, then you can do whatever you want with it, which I find it interesting. But there was no Pisha pastori strain in it, and then you have to agree that you will never use it in India. So this was one way to find the strain. Another way was in other commercial solutions. So at one point, Atom, which became DN 2.0, was offering a Pisha pastori strain, and they claim it was open source. But basically, when you look at the MTA, it was not really open source the way I see open source. It was that you don't have to pay commercial fee, but in the MTA as well, you can't share the strain that you have modified. So you have part of it which is good, that you don't have to pay a commercial fee, but then you can't share the strain. So for open insulin, it was an issue. I think it's an interesting example because it's also an example of at one point where there was, like highlighting the paper I show here, that there was some kind of push from the industry to say, oh, maybe we need more open source even in synthetic biology. But it seems to me, and maybe people after my talk can correct me if I'm wrong, but it seems to me that there is no motivation now with the inside the industry to try to do open source anymore. If you look at the DNA 2.0 website, there is no mention of open source, and the only open source strain or material that they share is the one they did in highlighting this paper in 2013. So to me, it kind of speaks to that we can't really rely on the industry right now to do open source stuff. So we have to find another way. Um, yeah. Oh yeah. Sorry. It's what I was saying. So what did they say in the? They say there is that you don't have to use it for license, commercial license, but you can't share it basically. So another strategy. So how we solve this problem? So there is different strategy. One of the strategy that we have seen is to try to negotiate with existing biological resource. So for OpenPCR, for example, they negotiate with a Belgium biological resource, and they look at a strain which was before GS115, and then they say, okay, we will take this strain, negotiate with the biological resource, and then modify it to be able to use it. So it's one way, but then when you look at the on the MTA, when on this biological resource, the Belgian biological resource, then still you can't really share the strain, the modified strain. So it's license free, but you can't share the modify freely. So you have to negotiate case by case with the biological resource. I think it's a so I had Jean here because I think it highlights that one of the strategy you can do is try to push existing biological resource to modify their practice, and I think it's a good example of that because they know they are you can't deposit you can deposit plasmid with open MTA with Agin, but I think with the biological resource with strains, you can try to push them to allow open MTA, but at the same time, I think what happened for all the strains which are already existing. So I think it would be complicated to revert the license. So the strategy we choose is basically to start from scratch and say, okay, we will not do the negotiation with the biological resource. We will just try to find a way to find strain and maybe redo a lot of the work that have been done of characterization of strain and which one are high expressing and so on, and then we will do that. And so basically, you are on the right. You have a picture of me in my family house backyard, and I just took some sample, put a bit of kanamycin, and isolate strain. And so far, we isolate 9 strain that look like yeast. And for some of them, we did a bit of barcoding to confirm that it was yeast. And then we say, okay, if we want to be able to use it and to engineer it to express protein, then the best is to have whole genome sequencing. So we send the genome into to a platform, and now we got some results, and we are hoping to analyze the genome soon and be able to share the strain and shares the genome with it. Then people can take the strain and then play with it to produce protein. So our goal after that would be to use it for the Open Insulin project, of course, but also for Enzymethic to use it to produce enzyme that we can sell in the market. So the main one would be trypsin, caxin, and protein SK. And the plan is to share the strain. So ideally, what we want to do is to make our own biological resource. So my understanding, and this is, say people can correct me after the talk if they know more than me about this, is there is no strong regulation, at least in France, to do your own biological resource. There is a guideline from the OECD of good practice that I think we want to abide to, which is basically about really practical logging, measuring, I mean, recording every time you send the strain to somebody and all that stuff, and the way they give indication about the way you should store them, the way you should ship them, and all those things. But as this is something that we can do, and then the goal would be to have some kind of platform when people can say, okay, I want this strain, and then we can send the strain. And ideally, in the really long term, and maybe when we have a bunch of lawyer in the team, the goal would be to be able to share modify strain. And there is a notion that why it's different from yeast and from E. coli is with E. coli you can share the plasmid, and normally more or less when you do the transformation into expressing bacteria, then it will produce your protein in a yield which is similar to another transformation. With yeast, it doesn't really work that way because you do DNA integration. You can have a lot of variation when you do the DNA integration. So basically, when you have the strain and you develop the strain to be a high producer, so you screen, you find the one which I have producer, then the goal would be to save time for other people. And instead of saying, okay, you have this DNA, then you have this strain, you can do the lab work to do the screening, then you will waste time for other people to screen modify yeast. So the goal would be to be able to share the modified yeast which are high producer. The problem with that then there's a lot of regulation for GMO and how to share GMO, and it's a completely different ball game than only sharing unmodified strain. So this would take a lot of legal work, but I think this is really something that would make sense to as the sharing material goes, that you don't only share the tools, but you share something which will save a lot of time for other people. So now we talk about all the projects that we want to do with Enzymethic. So we will try to produce a lot of the open bioeconomy enzyme. So OpenVent, BSTLF. I didn't put all of them. So there's the 980 enzymes. So the goal would be to produce them, test them with all the equipment we have. I think we can do a good job to find good protocol and characterize all the enzyme that we are producing and doing and sharing all this information back and optimizing everything, that people who also got the enzyme from bioeconomy would have optimized protocol to produce those. And we are looking also at enzyme from natural source because it's faster in a way. So we are looking at lysozyme from eggs because it's really a classical one and it's really easy to produce. And another one would be papain that recently we are looking at because it's not that complicated to purify. And we think it could be a really good alternative to protein SK because it's an enzyme which digests protein in differentiated manner. So you can use it when you do, for example, DNA extraction, and then you can use papain instead of protein SK. So this is our goal. So ultimately, what we kind of see ourselves as is some kind of crash test for the all the open source element of it, because I think that if you don't go go until the commercialization of things that you don't really know if something is truly open source in a way, because you don't have how to say that if it's if you commercialize something, then there's an interest for other party to consider a lawsuit in a way, and if you don't have that nobody can say, oh, this is really open source, because it's kind of up in the air. So our goal is to do the cooperative produce enzyme, sell enzyme, and say, okay, nobody came at us, hopefully, so it should be safe, for other people to create another their own cooperative and then make their business also that way on this. So it's kind of we see ourselves a bit as a pilot. I mean, at least in Europe, because I know there is commercialization in Africa, but I don't. To my knowledge, I don't think there is equivalent of what we are doing in Europe. Okay, so I would like to thank the amazing community lab who host me when I was in the US, Counterculture Lab and Bugs. I want to thank BioBrick Foundation for all the work they have done. I think, like, for the Open MTA, so I think this is kind of the really foundational work that allow me to even believe what I'm doing is possible. I thank Reclone for hosting me and for all the work they are doing. And I also want to thanks Ecosoli, which is incubator that we joined recently, and they help us for the business side, which I think we really need. And now I'm opening up for the question.
Awesome. Thank you. Thank you so much for sharing. Especially, sharing some of the background. It's amazing to see also how many collaborations you had. Yes, we have already some applauses on Zoom. So yes, now I'm going to pass to Yankee to help with the questions. As always, you can raise your hand on Zoom or send your questions on our chat, and we'll pass them to Ian. Thank you.
Great. Yeah. Thank you, Yann, again, for introducing to us about Enzymethic. I mean, it's definitely fairly risky, I guess, being the pilot's cooperative.
Yeah.
With what you and Louise are doing. Are there any specific kind of challenges that you encountered that you could give advice to other people about in future if they want to start their own?
Yeah. For us, I think it really depends where you are and how the system works. But for us, one of the big issue we had is the regulation system, because in France, at the moment you have a GMO in your company, then you become a registered company. I don't know how to translate it, but it would be called something like that. And it means that you have really strict rules to abide with concerning fire regulation and a lot of things. So finding a space was really complicated. And the issue is, at the beginning, we wanted to find a laboratory space in incubator, but because we said, oh, we want to do open source cooperative system, they were not really interested in helping us, because the metric they use for innovation or for company they want to help, no, the metric they used for company they want to help is innovation, and for them innovation is patent. So we got a lot of closed door because we were saying, okay, we want to do open source cooperative. So this was one of the big complication for us. So we had to do everything a bit on our own and find our own space and negotiate with the how to translate the environmental department of the place. So yeah, it was this was a big issue. Yeah, other than that, yeah, I don't know. Maybe with the question, I will have more inspiration.
And I'm sure it's a lot of challenges finding space,
Yeah.
facility, and then apply
Yeah.
complying with regulations. I'm gonna pass on to Pablo for his question. Pablo, feel free.
Hello, everyone. Thanks for the talk. It was very interesting. I have a question. Do you have any particular reason for choosing yeast or pichia instead of bacteria or the other kind of expression system?
Yeah, so for the insulin expression, I mean, you can express, for example, glargine, that it's one of the main form of insulin that is sold in the market. If you express it in bacteria, then it will go into the inclusion body, and then you have to do some kind of refolding process, which is really not, friendly for.
Yeah, cost, time consuming, etc.
Yeah. So the best way to do it is to use pichia pastoris and to secrete it into the medium. And actually, there is a way when you can. I mean, it's patented, but there is a way when you overexpress the caexin, which is a membrane protein, which is inside the Golgi apparatus, and it will digest your pre-insulin, and what you secrete is pure insulin. So it's a nice system, but since it was patented, we tried to find a way around this. But yeah, for the yeast specifically, the main decision was from that, and then I mean, I like working with yeast, and I would love to see a open yeast strain, and
Understand.
Yeah.
And have you thought about the possibility of having problems with the overglycosylation issue with yeast?
Yes.
Or
No, it's a real issue. I think for insulin there is not so many sites of it, so there is some, but there is not so many. But then you have to check it afterwards. But yeah, it could be an issue. But this is something you have. If it's an issue, you have to engineer, I think, the yeast strain to reduce and then check if it's not the engineering, it's not patented. It's always the same loop. But it's right now in the industry for insulin, the most of it, I think, it's produced in yeast, not necessarily pichia. There is still some with Cerevisiae, but yeast is a big producer. So they found a way to avoid the glycosylation aspect.
Thank you.
Maybe there is some stuff they don't say, but.
Okay. Thank you very much.
Sure.
Thanks. And Eric, you had a question?
Yeah, thank you. I got a few comments, questions,
but
I'll try to make them quick. One, I just want to say that I can sympathize with the issue that you had with with your strain initially, with finding out that you couldn't use it afterwards. We've had similar issues in the past. One quick thing that I would just say, you mentioned on the slide that plasmids are easier in some ways, and that is true to an extent. I will say that because every country has a different definition of what a GMO is, we do actually have a number of countries, through Agene that will, because we usually distribute things in bacterial, for those of you who ordered from us, we usually send things out in E. coli, but there are some countries that consider bacteria transformed with plasmids GMOs. And so we cannot send them bacteria, and we have to send them DNA. And that is just a quick clarification. In terms of the MTAs, I think if I understood you correctly, you mentioned that it's hard to go from an MTA backwards to, or not backwards, but from a regular MTA to an open MTA. And that is true. So that is something for those of you who are familiar with Agene, we do have an MTA system, but we do allow people to deposit things with us as open MTAs. And we have found that it's very difficult to contact previous depositors and convince them to switch from an MTA to an open MTA in general. The one thing I will say about MTAs, and this is more maybe finally getting to a question as opposed to a comment, is having attended a number of biosecurity policy symposiums and seminars and like that on behalf of Agene, that is the one of the few benefits of having an MTA is that it leaves a better paper trail, basically, for whatever your biological reagent is. And that's something that's becoming increasingly important, at least within the US. So I guess my question is, has that issue of biosecurity around having an open resource like this ever come up? If somebody can order something from you and then give it to whoever they want within France, is there a risk that you'll lose track of who has what? And I don't know if that's as big an issue in Europe or not.
Yeah, I mean, it's an interesting question. I think we will follow. Our goal is to use OpenMTA. So if we use OpenMTA, there will be some tracking which go with it. I'm not really opposed to it. I'm always finding the scare about biosafety, biosecurity a bit overblown. But I mean, it's my own opinion about that. You know, but I'm a biologist, so I'm biased, I think. But this is something we will have to figure out. I think, responding to your question, is how we can to whom we can share and what are the rules in this kind of thing. That if it has to be an institution, what you can recognize what is the which criteria is you need to use to define an institution. To me, those are a bit vague, and I think a lot of companies use their own criteria and their own, which is something I didn't mention in the talk, but I think sometimes it can create friction. Like if you're an independent researcher in the US, and then for some reason the company you buy material from decide that, oh, you don't respond to our criteria of institution anymore, we will not sell to you. And to my understanding, there is no strict rule about that. So yeah, my philosophy is more that we should share the maximum to everybody, but I think, but keeping track of to whom we are sharing it, you know. But the nature of OpenMTA, and I think it's a big argument that people can have against it, is at one point it's free to share to everybody afterwards. And so some people we share it following, keeping the paper trail, but some people we not. And yeah, at the end you can't really control what people do with it afterwards.
Well, yeah, there's two sides of that, I think. There's not being able to track who the people who get it from you share it with.
Yeah.
And that's the loss of the paper trail. The harder thing, and again, I'm speaking a little bit from Agene's own experience here, is because there's no restrictions on that, there's nothing stopping, you know, a commercial company from taking that and just deciding to make a product with it themselves if they order it from you and selling it. And basically, if they have the sort of the market presence, if you will, to sort of promote that and do that, they can. And then your own endeavor is sort of overshadowed a little bit, particularly if you're trying to like create revenue. You know, if you're, I think looking at your website, you charge a small fee for some of this stuff. And if somebody has a bigger name, whatever out there, they can get that out there, and suddenly people aren't getting it from you. They think that the only place to get this thing is from this big company that's charging 3 times as much money. And that's a tricky thing to deal with, I think.
Yeah, it's interesting. It's kind of a business question. Is it okay if you do things truly open source, then what prevent company to do it? And my understanding of and this is stuff that can be disputed, but my understanding of the market of enzyme is crazy overpriced because there is an oligopoly system. So you have few big players, and they can really charge a lot. So the idea, if I produce something which is already in the market and I produce it less, yes, they can use my material, but they're already producing it. So for them, the goal is not to replicate what I'm doing. The goal is to shut me down, basically. So then there's a lot of different strategy you can imagine, which is buyout, which is lawsuit, which is, but I'm not afraid they will take my stuff to do things, because they already have that stuff. So it's a different logic, but it's not necessarily crazy better, because then you can say, okay, if they decided to put you in court, then what you gonna do? Because they have way more money than you, so there is a lot of risk associated. But the risk of them using my stuff and making the product with it, it's not something I'm really scared of, to be honest.
Yeah, Scott, go.
Yeah, so actually, on Monday, I had shipped from the UK to me a strain of Pichia that has been engineered just like Open Pichia. And this is released under the Open MTA. So I haven't yet received it. It was held up in customs yesterday. It was supposed to get delivered yesterday, but it's been released from customs, and I'm checking this morning just to make sure it gets on the truck, but it should arrive today. So this is an alternative to Open Pichia. It's truly sent to me under the Open MTA. And it's based on NRL ARS culture collection yeast strain. Essentially what I was working towards doing myself. I'd already cloned out the homology arms and was preparing to engineer the same strain, but somebody beat me to it. So yeah.
Oh, great.
That might save you some effort, but it'd be also great to actually see new strains.
Yeah.
My one question, were you planning to delete the Hawk1 gene as Open Pichia did? So what kind of strain engineering are you thinking about?
Yeah, I think so. This one is the one for helping for transformation.
Yes.
Yeah, I think it would be great. I'm kind of my logic would be maybe I try to do transformation first and see if it's working well, and then if it's not, maybe deleting it. But I need to look more in detail for the open all the modification they did, because I didn't look everything that they did.
Yeah.
But there's yeah. Sorry.
If you'd like, I can share my homology arms. I've had them whole plasmid sequenced. They're Q5 PCR from the NRL strain, and most likely your strain would be similar. It'd be interesting to see, how divergent it is compared to the other strains, the FAF collection.
Yeah, we have some preliminary genome data right now, but there was some issue, I think, in the platform that you use. So I don't know how much coverage we have of the genome of the strain we have, but it's ongoing. Maybe we negotiate with the platform to do more sequencing. We see. But once we have it, the goal is to share the strain, but associated with the genome data, that people can really play with it.
Okay.
Yeah, definitely. I mean,
Yeah.
with Scott, I don't think it's necessarily they beat you to it. It's more that you are now collaborating, I guess.
Yeah,
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 yeah.
A lot of
and
interest.
yeah, exactly. And I think the more diversity that's out in the ecosystem, the better.
Yeah, definitely. I mean, I probably should have made this link earlier, but happy to connect with you all guys together with the opium pickier stuff, because I mean, Scott with his open yeast collection and yeah, and Jan with the picky and everything. So it's we can definitely make a small corner on the Recon forum where you can discuss this
Yeah.
more directly, I guess.
Yeah, I would be happy to.
So the strain that I'm receiving, they haven't yet finalized the name that they want to call it. They were suggesting opener Pickia as opposed to open Pickia.
Okay.
Uh,
Open pick, yeah.
Yeah, opener. I suggested open Pfaffii, but yeah, that's what I would have called my strain. But yeah, I like opener Pickia.
I didn't mention, so I'm going putting my businessman hat. We had a small questionnaire that we did for the to ask for people to fill and to have some insight of which enzyme they use and if they would be interested on buying from us and yada yada. So I will, if you don't mind, I will put it on the chat and then people can fill it if I find the link.
Feel free to put it on the chat. Feel free to post it in the forum as well to reach a wider audience than the folks we have today. But yeah, anything we can do to support one another in this community is always welcome, I think.
Thanks.
So I'm just going to stop the recording now, just to give so one sec.